Hello, everyone. My name is Eric Wengert, and I'm a graduate student at the University of Virginia in the lab of Dr. Manoj Patel. And in my research, I study SCN8A encephalopathy, and I'm very thankful to be given the opportunity to present for the third time a talk that I've given before called SCN8A 101, which goes through some of the basic science behind voltage-gated sodium channels and neurons and brain function. And it's just a kind of intro level discussion of some of the topics that are related to SCN8A encephalopathy. And so I'm happy to do that again this year. And I thought it would be good, since we're in a virtual format, to pre-record the lecture and distribute that so that you're all able to watch on your own time, and perhaps a watch ahead of time for the virtual meeting this year, and then be able to come with questions. So without further ado, this is the SCN8A 101 talk that I've prepared. One note I always like to mention in the beginning is that one, I'm not a clinician, I'm not a medical scientist, so I can't speak as much to the clinical aspects of SCN8A. I am able to speak to the scientific research, and so that's what I'll be doing today. And additionally, I always think it's important to highlight that I don't come to you as an expert in SCN8A encephalopathy. In the research community, we are confident the real experts are you as the families, that you know more about SCN8A encephalopathy than we do. But the piece of the puzzle that we can bring is to talk about the scientific research and what is going on and some of the directions that we're interested in pursuing. And so I appreciate your willingness to allow me to give this talk, and I hope you find it useful. So with that, we'll get started. So just a little outline that outlines what I'm going to talk about today. We're going to start off just really quickly talking about what the brain is, which I'm calling here the highly complex master organ of the nervous system. And just a brief, I'll mention briefly what the brain is and what it does to give a sense SCN8A and set the stage for talking about neurons, which are the specialized cells of the brain. And then I'm going to zoom in even further and talk about action potentials, which are the electrical signals that neurons use to communicate. And then lastly, we'll finally reach our discussion about voltage-gated sodium channels, which are the drivers of action potentials, and of course are relevant for SCN8A because SCN8A is the gene that encodes one of these voltage-gated sodium channels called NAV1.6. So that's our roadmap for today. And so let's begin at the brain. Think about what the brain is fundamentally. And I want to stress to you that the brain is kind of like a computer in that it's an input-output machine. It takes in information from the senses. So from the external world, you can sense light or sound. So it sends that information into the brain processes that, and then it decides on some action to initiate as an output. So this would be movement or speaking or thinking. So the brain really does have this direction of information flowing from senses to the brain, which is processing, and then it decides on some action or output. So it's an input-output machine. Now that's simple to say, but it's very difficult to actually understand precisely how the brain does this. And one of the reasons is because the brain is composed of 100 billion neurons and other cells. So there's a lot of complexity built into the brain and its structure. And again, structure, complexity relates to complexity in the function of the brain. And lastly, it's not only complex in terms of its parts, but the brain is actually dynamic in that it changes itself over time. So it's constantly adapting to its environment. Call this sometimes learning and memory. Learning and memory is actually one of the ways the brain can change itself. But of course, you also see it just change over development from one time to the next. And so these I just wanted to give as examples of why it is extremely hard to understand precisely how the brain works, but our goal as researchers is to understand perfectly how the brain takes in information, how it processes, and then how it initiates output. If we fully understand that cycle, we would fully understand the brain. Within the brain, the processing or the computation that the brain does is accomplished by these specialized cells called neurons. And again, these are any specialized cell within the nervous system, those are called neurons. And they have three main structural components that map on to that directional flow of information that I just presented in the brain as a whole. So within individual neurons, you see a region that corresponds to the information input. This is called the dendrites. Secondly, you see the cell body, or sometimes you hear soma, which is where that information is integrated. And then lastly, the axon, which is the output of the neuron that either sends the signal to another cell or to the body, to like a muscle or something. And so again, just as with the whole brain, individual neurons have an informational flow. And what makes neurons unique is that they are electrically active or excitable. This means that they signal using the flow of positive and negative charges. These are sometimes called ions. So the flow of ions actually accomplish the signaling that neurons are using to communicate with one another. Here's just a quick example of a couple different types of neurons that were reconstructed from various animals, rodent models, as well as I think a few human cells in here. And this is just to highlight that when I show you a cartoon that I've made of neurons, that I'm highly simplifying this. The truth is we still don't fully understand how even an individual neuron functions completely in terms of input and output. And so for that reason, we are still a long shot from understanding how the brain functions as a whole. You can see that these neurons are very different, very diverse, and highly complex even within themselves. And so I just want to stress that today, as I go through things, I am simplifying a bit just to help with our discussion today. But in general, we're still very active. These are active areas of research to try to understand how individual neurons function. And so I said that neurons use electrical signaling to communicate. And it's helpful, I think, to consider why the nervous system uses electrical signaling rather than a different type of signaling. And the key takeaway from the nervous system is that electrical signaling is the fastest type of signaling in the body. In that, in using electrical signals, like what we're going to talk about, we're going to talk about action potentials. Electrical signaling is so fast that it allows the body to take in sensory information and respond on the sub-second time scale. So if you saw a bear threatening you, you'd be able to see it and respond in an extremely fast time scale. And this is important for survival. Nervous systems were evolved to help us survive in our environment. And so you need, in order for a nervous system to function properly, it has to be extremely fine-tuned because it's acting in extremely fast time scales. The fundamental unit of this electrical signaling, the individual unit of processing, is the action potential. And so we're going to spend a bit of time talking about what that is, because it is very important to nervous system functioning and it relates to voltage-gated sodium channels. Essentially, what an action potential is a quick spike in membrane voltage. If this term voltage gives you negative flashbacks to high school physics, don't worry. Just think of it as the electrical energy of a cell just has a quick blip. You can see like here's another example as well as this one. And I just showed these examples. They're all on the same scale just to highlight again the amount of diversity within cellular function. So it's easy to talk about action potentials being a singular thing, but the reality is cells are using, even though they all fire action potentials, the individual action potentials are highly unique, and we're still trying to fully understand all of the ways that the action potential is used for signaling. But for today, we will zoom in and focus on the action potential, which is just this quick spike in membrane voltage that neurons use to communicate with each other. And that's thinking about single cells, but the reality is that neurons function in networks. We use this term to talk about how cells are signaling with electrical signals, and that signal can be carried through multiple cells. So you see in this little clip how electrical signaling is flowing through many neurons to eventually accomplish some task. And so this again adds complexity to the system, which matters because it's difficult to understand experimentally. And the reason I want to mention that networks are involved in the brain is because seizures fundamentally are due to hyperexcitable and hypersynchronous networks. So when the brain has networks that have too much excitability or too many action potentials, and then they eventually all are being excitable in the same time scale or hypersynchronous, that can lead to a seizure. And this is important because that means that the brain has to maintain a very precise balance of excitability. Obviously, you can't have too little excitability, or else you'd find it difficult to function and fully take in information, process, and respond to the environment, as well as you'd have difficulty in learning and memory. So you have to have enough excitability, but you also can't have too much, or else the networks will break down into hypersynchronous activity and you'll have a seizure. And so the brain is tasked with a very, very difficult challenge to maintain excitability. And sometimes I think of it like walking this tightrope that the brain is maintaining a very nuanced balance of excitability. And so in thinking about neurons and networks paired with what we've already gone over, I wanted to give you three major aims of epilepsy research. And these are kind of categories that I use when I'm thinking about when people are studying epilepsy as in scientific research. What are the kind of questions, big questions that they're after? And I think in epilepsy, three of the biggest questions are, one, how do neurons become hyperexcitable? So just trying to understand what are the ways in which neurons have too much excitability. Secondly, how do networks become hypersynchronous? So what are the mechanisms that allow neurons to fire together and have action potentials all happening at the same time? And then lastly is the treatment aspect of epilepsy research, which says, how can we restore balance to the brain? That's this piece over here. How can we restore balance to the brain in terms of excitability, not having too much and not having too little? And those are some of the, again, the major aims of epilepsy research, and those are the major aims of what we do in the Patel Lab, trying to understand hyperexcitability, hypersynchrony, and ultimately, how can we restore balance in the treatment of epilepsy? For the next phase of the talk, I want to go over the action potential in a little more detail. And where I'm going with this is we're setting up our discussion to understand how voltage-gated sodium channels contribute to neuronal excitability, and how when they have mutations, as in SCN8A encephalopathy, it affects how neurons function and leads to epilepsy. So again, I think I've already mentioned, but just to say again, action potentials are the language that neurons use to communicate with each other and with the body. And an individual action potential is this spike in membrane voltage over time. And it happens really, really fast. This is zoomed in on milliseconds time scale. And each action potential can be broken down into three parts. The first is this resting phase shown here in green, where the membrane is low and pretty stable. The second phase is the rising phase, where it rapidly shoots upward, followed by the falling phase, in which the membrane potential peaks, comes down, and returns to normal. So again, resting phase, rising phase, falling phase. kind of makes sense. And then of course, after the falling phase, it goes back to the resting phase and the cycle can start again. So this is these are the phases of the action potential. Each action potential has these phases. And so how do we even run an action potential? We have to set the stage for it. Since the action potential uses energy, you kind of have to battery charge the system. And so now I want to talk about this battery charger, which is the cells have throughout their entire membrane, this on the outside of the cell, this little battery charger channel called the sodium-potassium pump. And it's going to be critical for running the action potential. And so here again, I've zoomed in here in red on the cell membrane, and we have many different ions, both on the outside of the cell and on the inside of the cell. And I've shown them here colored, yellow for sodium and green for potassium. And when we run the battery charger, what happens is it pumps all the sodium on the outside and all of the potassium on the inside. You can see here. And this is important because we're actually going to use the energy that's stored in the ions being separated like this to be able to run the action potential. Now, just to give you an example of how to visualize this, think of it kind of like a dam where the water is extremely high on one side and low on the other. All you have to do is open up the possibility for the water to flow through, and it will flow through with a massive amount of force because you've created this gradient between the water levels. In the same way, when we open a channel for the sodium, it's going to flow through with a lot of force because it's very high outside but very low inside. And the same thing would happen with the potassium flowing outward. And so again, in the same way that you open channels and water rushes through with a lot of energy, the sodium-potassium pump pushed the sodium outside of the cell, and we're actually going to use that energy when we have the action potential. And the channels that sodium use to come back into the cell are voltage-gated sodium channels. So again, we set the stage by pushing sodium all to one side with the sodium-potassium pump. They're going to be allowed back through into the cell using voltage-gated sodium channels, and that is going to be the basis for our action potential. So let's talk about voltage-gated sodium channels. Again, these are channels that sit through all along the cell in the membrane, and they're going to allow sodium, specifically sodium, to pass from the outside back into the cell. But they're not just always an open channel. It turns out these channels have gates that determine whether or not the sodium can pass through. They have two gates. One is an activation gate, and one is an inactivation gate. And using these two gates, opening and closing, we get three distinct channel states that are really, really important for the action potential. The first one is a closed channel. It has the activation gate is shut. Sodium can't pass through a closed channel. When the activation gate opens, then you have an open channel. Again, both gates are open, and that way sodium can pass through. It turns out though, with an open channel, it's only stable for a few milliseconds, and then in the process of inactivation, the inactivation gate closes, and sodium can no longer pass through. So again, closed, opens, activates, and goes to the open channel state, and then very quickly it inactivates, and you have a cycling of channels through these states. And the cycling of voltage-gated channels, voltage-gated sodium channels through these states is important for the action potential. So in the same way that we have three channel states, and I told you earlier that we have three action potential phases, it turns out that the voltage-gated sodium channels run the action potential, and that the phases of the sodium channel correspond to these three distinct occurrences during the action potential. So during the resting phase, we have the voltage-gated sodium channels are closed. when they activate, sodium rushes in, and that positive charge from the sodium is what drives this spike in membrane voltage, because it's a positive charge going into the cell means voltage goes up. However, it inactivates and the action potential experiences the falling phase. The membrane potential comes down back to rest as the sodium ceases to flow in. And so again, cycling through these 3 states of voltage-gated sodium channels is critical for firing an action potential. And any small deviation in the cycling that channels are normally supposed to do actually will have very, very strong consequences in terms of the action potentials that they send. And that is what happens in the case of SCN8A encephalopathy, where a mutation actually changes the way that sodium channels progress through these states. And we're going to talk about that. We've gotten pretty far through the talk without really mentioning much about what SCN8A is. Well, SCN8A is the gene, which is a term that uses a sequence of DNA that codes for the NAV 1.6 channel. And so the simplest way to think about this is that SCN8A is the instruction manual that a cell uses on how to build the NAV 1.6 channel. And when there are mutations in SCN8A, we're talking about that the genetic sequence is altered in some way that changes the directions for building the channel, such that when the channel gets built, there's a small alteration in the shape or structure of the channel, and that changes the function of the channel. For example, in scientific research, we often use a mouse model that has the N1768D mutation. And this is a shorthand for saying that in the 1768th position, we typically would have an N, but a point mutation has changed it to a D. And this single change changes the way that the channel functions in a way that that leads to epilepsy, leads to spontaneous seizures. And so again, the SCN8A is the gene that codes for the NAV 1.6 protein or channel that actually does the function that drives action potentials. And so what makes NAV 1.6, again, the product of SCN8A, what makes it unique? I want to give you just 3 examples from my own research that highlight how NAV 1.6 affects how neurons function in a way that is relevant for neurons becoming hyperexcitable and contributing to epilepsy. And 3 of the important aspects of action potentials that are determined by NAV 1.6 are: 1, when a cell determines to fire an action potential; secondly, how many action potentials are actually generated; and third, the shape of that individual action potential. All of these are strongly affected by NAV 1.6. And so I'm just going to show a couple examples of recordings that I've made from neurons from mice that either have a typical NAV 1.6, we call it wild type, or a mutant, an NAV 1.6 channel that is generated from an SCN8A gene that has a mutation. And if I do the exact same experiment, in this case, I want you to appreciate that for this red recording, it initiates the action potential too early, both in time, so it happens to the left of the black one, but it also happens where the spike starts from a lower membrane potential. So you can think of this as mutant NAV 1.6 drives cells to initiate action potentials too early. Secondly, if I record and I stimulate neurons for this rectangle amount of time, you can see that they start to fire repetitive action potentials. If I do it in a neuron that has a mutation in SCN8A and has a mutant NAV 1.6, you can see there are more action potentials given the same stimulus. This is what hyperexcitability of neurons looks like. For given some input, the neuron has too many action potentials in response. And you can think of this kind of like a turning up of the volume of every single stimulus. The neurons are just dialing up the signal and it becomes too large. And lastly, it's not only that mutant NAV1.6 generates too many action potentials, but occasionally it strongly affects the shape of each individual action potential, which is really important because since action potentials are the language that neurons use to communicate, when you change the shape, it's like a bad misspelling of each spoken word. It begins to change the meaning. And so I've just zoomed in on an example here, again, with the exact same stimulus. In a typical condition, you have just a single spike, it goes up, and then falling phase comes back down to rest. But in the mutant neuron, you have the rise of the action potential. But during the falling phase, it actually reinitiates a little spikelet and kind of has a very abnormal burst shape compared to the typical action potential. And again, this strongly affects the way that the neuron communicates with other neurons and with the body. And that changed signal is likely important for generating seizures in the brain. So just to summarize what we've gone through today in SCN8A 101, first we started out thinking about the brain. And even though we can think of the brain as being an input-output machine, it's extremely complex. And it uses electrical signaling to quickly and very rapidly take in sensations, process information, and execute actions. That's what a brain is for. And the brain uses neurons, which are special excitable cells that communicate using action potentials, these electrical blips that send information to other cells or to the body. And these action potentials, spikes in membrane voltage, are driven by voltage-gated sodium channels. These are specialized channels that initiate and propagate action potentials in neurons. And lastly, SCN8A is the gene for NAV1.6, which the function of NAV1.6 is critical for many aspects of neuronal signaling. I'm just again showing here that it affects things like shape, how many action potentials, and the timing of action potential. And so these changes, as a researcher, we are trying to understand how SCN8A mutations change neuronal excitability, leading to hypersynchronous neuronal networks. And ultimately, we want to know how can we restore balance in the brain to treat epilepsy and SCN8A encephalopathy. I'd like to end by acknowledging my team. I've listed here the names of people in the lab of Dr. Manoj Patel, and each of my colleagues is passionate about understanding SCN8A encephalopathy, and I hope that I hope it brings you comfort to know that there are groups of people who are passionate about understanding how SCN8A functions, and I can attest to all of their hard work as well as mine, and we're just thankful to be a part of this community and serve in whatever way we can. And so with that, I'll end. I appreciate you giving me the time to give the talk again, and I hope you find it helpful. If you ever have questions about any of the content, one, I'll just look forward to answering them when we're together virtually for the roundtable discussion. But if you do want to reach out to me, you're welcome to send it to my email. I'm happy to help in whatever way I can. With that, thank you.